So hello guys and welcome back to Excelsior Academy. So today we'll be starting a new topic as cell cycle. So cell reproduction, it perhaps is the most fundamental characteristics of cells, and all cells are reproducing by dividing in two. So we know that with each completion of a cell division, we are going to get two daughter cells, right? Now these newly formed daughter cells can themselves grow and again divide, given the condition that they have got proper nourishment, okay, or proper nutrition available, resources available, then they themselves can divide, giving rise to a new population. So the division of these cells, it must be carefully regulated and coordinated with both cell growth, DNA replication, in order to ensure that these these cells are containing the intact genome. This is really important. Otherwise, what will happen? They give rise to cancer cells. Now, in the case of the eukaryotic cells, progression through the cell cycle is controlled by series of protein kinases. And today we'll see, like in the case of the cell cycle, so many kinases are involved in order to regulate the cell cycle. In higher eukaryotes, these cell cycle machinery itself is regulated by the growth factors, which control the cell proliferation, which allow the division of the individual cells, and all these are coordinated with the needs of the organism as a whole. Okay, so aisa nahi hai ki like it is growing in this particular area properly and but not in the other region. It is well coordinated for the entire organisms. Now we know that if something goes wrong in the cell cycle regulation, it will result in the cancer cells. So what you will see is that in this lecture series, that anything goes wrong, okay, anything, even a single mutation, eventually it is going to lead to the cancer proliferation. Okay, now coming to the cell cycle. So there are 4 phases in the cell cycle, in which the cell growth, cell division, cell replication, distribution of the duplicated chromosome to the daughter cells, and the cell division is happening. Now cell growth means what over here? That is, see, when a cell is gonna divide, right? Before that, everything needs to be duplicated. Correct? Now, before that, before DNA duplication, you need proteins. The cell membrane needs to be expand, right? So more lipid synthesis, more protein synthesis will take place. After that, only DNA replication take place. Then distribution of the duplicated chromosomes. So this also we know. So when this at the metaphase chromosomes are aligned, so one sister chromatid should go in this direction, the another should go in the another direction, in the opposite poles, and then the cell is going to divide, right? Now, in the case of the bacteria, cell growth, DNA replication takes place throughout most of the cell cycle, and duplicated chromosomes are distributed to daughter cells in association with the plasma membrane. Now, in the case of the eukaryote cell cycle, it has got 4 discrete phases. It is synthesized during one phase of the cell cycle, DNA, and the replicated chromosomes are distributed in the another series of events. So now we know, right, that there are 4 phases in the cell cycle, and at the last phase, that is mitosis phase, the DNA is getting divided. Okay. Now, animal cells are used normally, to study about all these things. So the cell cycle is divided into 2 basic parts, namely mitosis and the interphase. Now, in the case of the mitosis, so जब पूरा का पूरा cell आपका 24 hours में divided है, like it is taking 24 hours to divide. Okay, so usually your normal animal cells, it is taking 24 hours, right? Now mitosis out of 24 hours is around 1 hour only, and interphase is 23 hours. Okay. So mitosis, as we know, it is the dramatic stage of the cell cycle. It corresponds to the separation of your daughter chromosomes and the carrying out the cytokinesis. Okay. Now, As we say, mitosis and cytokinesis only for 1 hour, rest all time is spent in the interphase. Okay. Now, during the interphase, the chromosomes are decondensed, distributed throughout the nucleus. So, the nucleus appears morphologically uniform. And at the molecular level, interphase is the time during which both cell growth and the DNA replication is happening. It's very simple topic. It's just that once we start talking about the proteins, little complexity will increase. So this is how the 4 phases of the cell cycle is divided. So you have here mitosis, G1, S phase and the G2 phase. Okay, so here we see that mitosis is around 1 hour and rest all is 23 hours. So you will see that 23 hours of the interphase is divided again into 3, G1 phase, S phase and G2 phase. Okay. Now, in the case of the G1 phase, so G here stands for the gap 1 and gap 2. Okay, so G1 ho gaya aapka kya? The phase which is followed by the followed by the G1, which is between the interval gap between the mitosis and the initiation of the DNA replication. So here in the G1, the cell is not replicating its DNA. Okay, but it is metabolically active. So any idea what does it mean being metabolically active? Yes, Chandni. Anyone? Krishita? We are saying that the cell is not dividing or DNA replication is happening.
Yeah,
But
the genetic material and everything is getting produced, but the division is not happening. All the enzymes and everything is getting produced, but the cell is not dividing.
no, but we already saying that it is not replicating its DNA. So it means the genetic material is also not getting,
Okay.
replicated. But we are saying it is metabolically active. So what is that thing which is required before DNA replication?
The proteins.
Yes, correct. So those are the proteins, the lipids, which is required in order to make the cell ready for it. Okay. So for example, DNA polymerase, those transcription factors, replication factors, which is required for your replicating the DNA. So those are required and those are getting, transcribed and translated at that time. Okay. So those proteins are getting ready. Now, G1 is
So,
followed by
ma'am,
S. Huh.
so the helicase and everything, that means the enzyme is also getting produced, right? The
Yes,
helicase
yes,
and everything.
yes.
Uh-huh.
Okay. Now, G1 is followed by your S phase during which the replication is taking place. Then comes the the completion of DNA synthesis, which is followed by the G2 phase, and during which the proteins are synthesized for the mitosis. Okay. So each stage is important. So G1 for preparing the cell for your DNA replication. Then at the S phase, the synthesis is happening, and at the G2, the proteins which is required for the mitosis is getting ready. Okay. So now you know, like for mitosis, what all is required, all your motor proteins, the cytoskeleton proteins and the proteins which is required, which, keeping your chromosomes together. And when the division is happening, those proteins need to be, the separase, securin, which is present, they need to be removed. Okay. So the duration, like how each phases of the cell cycle is divided. So G1 out of 24, G1 might take around 11 hours, S is around 8, G2 is 4, and M is about 1 hour. Okay. Now, for example, in the case of the budding yeast, it can progress through all the 4 stages of the cell cycle in only about 90 minutes. So as you see over here, even though the cell cycle has got all these stages, okay, most of, like mainly your bacteria, E. coli, it is taking only 20 minutes. Your budding yeast is taking 90 minutes, and whereas the animal cell, it is taking around 24 hours. So all these stages are important. It's just that depending on the size of the cell cycle time is also varying. Okay. Now cell cycle duration, for example, even shorter cell cycles, like even for 30 minutes or less, which occur in early embryo cells shortly after fertilization of the egg. Now, when the cell growth is not taking place, reason of that, these early embryonic cell cycle have to be checked. This is what is happening before during the embryonic development. So, जैसे भी fusion हुआ, okay, there's a fusion between egg cell and the sperm cell. What happens after that?
Ma'am,
Okay. So as I was explaining over here, so once these embryonic division is happening, so after the egg and the sperm cell is, they get fused, what happens is that there is no further lipid or protein synthesis happening. The same here itself, the cytoplasm is getting divided. Okay. Now once here is what is happening, just the DNA is getting replicated and mitosis. Replicated, mitosis, replicated, mitosis. Okay, but there is no further, what to say, like the protein synthesis or lipid synthesis happening. So usually what happens is when a cell is there, when it is growing, it grows double the size, so that when it divides, equal daughter cells we get, right? Which is like similar to the parental cell. अगर यहाँ पे ये 1 micrometer का cell है, तो आपके daughter cell भी 1 micrometer ही होंगे. But during this phase, what is happening is that they just keep on dividing. ठीक है? तो ये आपके 1/4 हो जाएंगे. And then again 1/8. The only intact is the genome, but the cytoplasmic proteins or cytoplasmic components are getting half and half with each division. And once they reach a particular stage, that 8 cell stage, then they start, having the development in the cytoplasm also. Like the cytoplasmic proteins or cytoplasmic components will also get double after that. Okay. So there are the some cells which cease division and divides only occasionally, जैसे कि हमारे nerve cells हैं, ठीक है? As needed to replace the cells which are lost due to injury or the cell death. Now, the cells exit G1 to enter the quiescent stage, that is G0 stage, where the cell is not dividing or proliferating, but it is just stopped. Okay, that is like the cell is not doing anything, it just stopped. That is like in few cases, like in the case of the mycobacteria, they undergo the dormant stage. Similarly, when the nourishment, nutrition, or the extracellular signals are not proper, okay, if it is not in favor of these cells to grow, then the cell will enter G0. Okay, Prishita, is this clear? Like what is G0 stage? Yeah.
Yes, ma'am.
Now, how do you determine the DNA content? So for that, we use FACS. Like what is FACS? Flow cytometer it is, okay, fluorescence activated cell sorter. Okay, so we'll study the principle of this FACS later in the, the methods in biology wala mein hum karenge. Now, the cells in G1 are diploid. So we know diploid means having 2 copies of each chromosome. So the DNA content is going to be 2N. So during S phase, the replication increases the DNA content of the cell from 2N to 4N. So in the case of the S stage, the DNA content will be known as the 4N. Okay. Now, DNA content then remains at 4N for cells in G2 and M, decreasing 2N after cytokinesis. Okay. So now look at the graph. This is important. So number of cells, and here you will see. So G1 में क्या है? You can see the amount of DNA per cell. So in G1 it is 2N. Then S phase में वो double होगा, तो G2 and M में आपके पास double the DNA content is there. So after M you will again go back to G1 जहाँ पे वो 2N हो जाएगा. ठीक है? Now the question is why in case of G1 the number of cells are looking more and G2 और M में कम दिख रहे हैं? What will be the reason of this?
Because all the content and everything is replicated, but the division hasn't taken place.
हाँ, so if I say number of cells यहाँ पे है, that is say the 100 cells are there, okay? These 100 cells will undergo and it will reach the stage of G2 and M, correct?
Yes.
Right? But then again the graph is always half. आप जब भी DNA content निकालोगे of your cell content, you will always get like this only कि number of cells हमेशा G1 में ज्यादा दिखेगा और G2M में हमेशा उसका आधा दिखता है.
Okay.
So the question is why? It is expected that अगर यहाँ पे 100 cell है under G1, so these 100 cells should also go to G2 and M, and यहाँ पे 4N है ना, so instead of having like this, we can have like this, something like this, where it is showing.
Checkpoints and everything.
No, checkpoints ensure that they enter the stages at correctly without any mutation carry forward.
Okay.
So my question is clear, right? Why
Yes,
this is
yes.
this much and this is half of it?
No idea, ma'am.
Okay, so it's very simple. It's never clear that they are either at the G2 or M. Okay, so we know that G2 and M, the content is 4N, and the moment the cell passes the mitosis, it is going to be 2N. So whenever you take this analysis of any cells, okay, we don't know how many cells are getting converted, undergoing mitosis. So क्या होगा? Number of cells having 2N is always high. G2
Okay.
plus M, we take it together because there is no clear-cut division. ठीक है? S, we just say that between 2N to 4N, it is the S phase. But there is no clear-cut division which says that अभी वो G2 में है या अभी वो M में है. As if you remember, G2 आपका कुछ around 4 घंटे का होता है. Immediately it is mitosis. Okay. So when we take the cells G2 or M, there might be few cells which undergo mitosis. तो क्या होगा? Number of cells यहाँ से decrease होके वो यहाँ पे increase हो जाएंगे. Correct? क्योंकि 2N तो यहीं पर ही आएगा.
Yes, yes, yes.
है ना? So there is no clear-cut division between G2 and M. So there are cells which might undergo directly, cytokinesis, and those cells का यहाँ पे refer होगा every time. So that's why जब भी ये question आए, keep this logic in your mind, okay? And same graph आएगा, same graph. You take any cell, same graph आएगा. Even I have done this experiment on my pancreatic cell lines, cancer cell lines, and even I got the same graph only. Okay, so कुछ भी आए कैसा भी आए, keep this graph in the mind and remember that this is G1 S G2 plus M और G2 plus M 4N रहेगा और यहाँ पे G1 आपका 2N रहेगा. So based on this only the numericals comes. Okay.
Okay.
Now coming to the regulation. So regulation is controlled by the extracellular and the internal signals. So first cell cycle regulatory point in the case of the yeast that will result in the progression from G1 to S. So कितने checkpoint हम देखेंगे? G1 to S में देखेंगे, S में देखेंगे, G2 में देखेंगे और एक M में देखेंगे. Okay, so first regulatory checkpoint was seen in the case of the Saccharomyces cerevisiae budding yeast and it is known as the start. तो स्टार्ट यहाँ पे क्या मतलब हुआ? इट इज अ फर्स्ट चेक पॉइंट, ओके, विच विल allow your budding yeast cell to enter the S phase after the G1. Okay. However, passage through start is highly regulated event in the yeast cell cycle controlled by the external signals. Now, what are those external signals? Nutrition available होना चाहिए, yes. Mating factors available होना चाहिए, yes. The correct cell size होना चाहिए, then only it will enter the S phase where the mother cell is gonna start having the bud formation. Okay, so this is the stage at which it decides, okay, whether the sufficient nutrition is available or not, and then only it will enter the S phase.
What is this mating factors?
Mating factors, this is like, again, it is telling whether, mating factors जो से हमने जो conjugation में नहीं पढ़ते हैं, in the case of the bacteria, where
Yes, yes.
है ना, F plus, F minus. So in the case of the
F
E's,
prime, F prime.
so those kinds of, you know, the factors or the other protein receptors and all, they are required. Okay, so that's when we say as the mating factors. It's not that, you know, it's not the sex-based mating factor. It is like for them, the answer to their cell कि हाँ मुझे अभी ready होना है for the budding. Okay. Now, similarly, in the case of the animal cell, it is known as the restriction point. Okay, so again, it depends upon the extracellular growth factors. In the presence of the appropriate growth factor, the cell will pass the restriction point and enter the S phase. Now, this repetition बहुत होगा भी यहाँ पे, ठीक है, कि कैसे और किस वजह से G1 to S phase में वो जाएगा. Now, once it passed through the restriction point, the cell is committed to proceed through S. Now, here also you have to understand one thing is that the growth factors or the requirements, okay, checkpoints to enter from G1 to S, if it is met, the cell will enter the S phase. After that, even if you remove the growth factors which is required, okay, to maintain the S phase, the cell is destined to enter the S phase. अब उसके बाद कुछ भी mutation अगर हो गया होगा, the cell will not go back to repair. It will be in the S phase and the DNA synthesis will take place. Did you get it?
Which you mean is once it enters the S phase, it won't go back. So it's
Yes.
a-
Yes.
Okay.
Yes. Even if the suppose it enters the S phase and यहाँ पे आपने chemical mutagen add कर दिया है, okay, which is not good for the cell, right? But since it has destined, okay, अब मैं आ गया हूँ S phase में, so it is going to divide, and whatever because of that chemical mutagen, any mutation is gonna happen, it is going to face that. Believing that there is another checkpoint which will make sure that this problem will be sorted out. Okay, so there will be repair happening.
That means when it enters into the S phase, it already has all the proteins, chemicals, and everything, and it cannot go back.
Yes, yes. एक बार signal मिल गया to enter the S phase, it cannot go back. Okay. Now, okay, that's the cell is committed to proceed through the S phase and rest of the cell cycle, even in the absence of further growth factor stimulation. यानी कि if you remove, so the growth factor which is required to enter into the S phase, now once you remove them, then also, see, it's a cycle, right? So it has entered the S phase. Now, obviously, to come back to the G1, it has to cross G2 and M, and then only it will come back to G1. ठीक है, so they are meant to complete the cycle. ऐसा नहीं कि S में आने के बाद वो वापस G1 में जाएगा, no. G2 में आने के बाद वापस S में जाएगा, no, it cannot happen. What can happen is only this one. Like G1 से वो G0 में जाएगा, G0 से G1 में जाएगा when you don't have the nutrition or the growth factors available. Okay, the cell gets arrested at that stage only. So on the other hand, sorry. On the other hand, if appropriate growth factors are not available, progression through the cell cycle stops at the restriction point. That is your G1 only. So G1 से वो G0 में चला जाएगा, where the cell can remain for long periods of time without proliferation of the cells, but they are metabolically active. यानी कि क्या, in order for the survival, what all is required, that will be happening. It's just that the growth and the division is not happening. But the cell is metabolically active. यानी कि glucose utilization हो रहा है to keep your cell, alive. Now, there are many cells in animals which remain permanently in G0, whereas others can resume proliferation if stimulated by appropriate growth factors or the extracellular signals. For example, skin fibroblasts. They are arrested in G0 unless and until stimulated by any wound. So, we know that in our case, once you reach a stage where your skin is not dividing, where your cells are not dividing, right? Then, unless until there is a cut, the fibroblast cells are going to remain in the G0 stage. Okay, so whenever this wound is there, in order to repair, then only there is a signal. Okay, now the fibroblast cells need to be divided so that the injury can be repaired over there. So those proliferation is triggered by the platelet-derived growth factor, which is released from the blood platelets during the clotting, and it signals the proliferation of fibroblasts in the vicinity of the injured tissue. Okay, so see, that's why as you see, we know that जैसे जैसे age होती जाती है वैसे वैसे आपके stem cells कम होते जाते हैं, correct? So these fibroblasts also, they have got the lifespan of around 40 generation. What does it mean? It means that one particular fibroblast will divide only for 40 times. And after that, it goes. It means what? Yeah, one fibroblast, this is going to divide in two. Okay. Then this is going to divide in two. This is going to divide in two. Correct? So the one copy which always have got, you know, the older proteins. Okay. This is also, you should understand that whenever a cell is dividing, there are always old proteins, old cell components and the new cell components are there, right? And जब cell divide होता है, if we say that they all are getting equal, old, new, old, and new. Now, among these two, which cell should die first because of the aging?
Old.
No, this is cell 1, this is cell 2. Old, new, 50-50 मिला है दोनों daughter cells को, right? So which cell should die because of the aging? कोई नहीं मरेगा because both are equal, right? But this is not the reality. Reality is that once the cell divides, okay, one of the cell is gonna get 90% of old and 10% of new. And here 90% of new and 10% of old. Now because of the aging, which cell is gonna die? This one.
90% old, yeah.
Okay, because they carry the old proteins. So this is the one which will die. So we know that this is again gonna divide. This is again gonna divide, right? So this after division, again, this particular cell is gonna have 90% of the old ones only. वो old इतना इकट्ठा होता रहता है over a period of time. Okay. Eventually, the cell is not able to repair itself. यानी कि since it's all the old proteins and all the old components are there, that is when the cell die. Okay. So it means what? हमने एक से चालू किया. Let us say that 2⁻40 generation में जो आपका first one था, which carried, you know, the oldest of all the cell components, now it's dead. Okay. So this is how the cells will, you know, start losing. So eventually एक time आएगा जहाँ पे जो last cell होगा, even that will have the, you know, 90% of the old components. Eventually it will also die. And that's how what happens is that over a period of time, then we don't have new fibroblast growth. It's just that जो पहले से present है, they all are doing their function. And eventually when we grow, you know, and then the बहुत सारे factors होते हैं for, you know, aging in a human. So did you get it? And this is true for all kinds of cells. Clear?
Yes, ma'am.
Yeah. Yeah, okay. Now the cell cycle regulation eukaryotes, although the proliferation of most of the cell is regulated in G1, some regulation is happening in G2 also. So one example is there in the case of the fission yeast. पहले हमने बड़ी yeast देखा था, ये fission yeast का है, ठीक है? Different phases of cell cycle is dependent. There is nothing given क्या? Okay, I don't know, something is missing. Here it is. Different phases of the cell cycle is dependent on series of cell cycle checkpoints, which will prevent the entry into the next phase of the cell cycle. Okay. So these cell cycle checkpoints are also known as the DNA damage checkpoints, and they function to ensure that the damaged DNA is not replicated and pass on to the daughter cells. So how many are there? So you have got in the G1, then the S, G2, and at the M phase. So total 4 checkpoints are present in any cell cycle. Okay. Now they function in G1, S, G2 phases of the cell cycle. Now let's go with the G1 checkpoint. So G1 checkpoint, it allows repair of any DNA damage to take place before the cell enters the S phase. So what is happening in the G1 phase? Before replication, if any damage is there, that should be repaired, because we don't want to replicate the damaged DNA. Okay. Now, second S phase, it not only provides the continual monitoring of the integrity of DNA, okay, but also provides quality control monitor to promote the repair if any error which occurs during the DNA replication. So we have seen like how in the DNA replication, how the checkpoints are functioning. कैसे कैसे अगर कोई mutation अगर introduce हुआ है, so they make sure that those are being checked. Okay. Now coming to the G2 checkpoint, so it will prevent the initiation of mitosis if the cell is containing the DNA which is not replicated properly or which has got the damage. So it will ensure that in G2 it gets corrected. Otherwise, what will happen? It will enter the mitosis. Now again in the mitosis we have got the last checkpoint. So spindle assembly checkpoint, it will monitor the alignment of the chromosomes on the mitotic spindle, ensuring that the complete set of chromosomes is distributed accurately to the daughter cells. Otherwise, that again at that stage also the cell gets arrested. Okay, so they try to repair it if they see that the mitotic spindles are not attached to the chromosomes properly, so the division will not take place and the cell is meant for the apoptosis. Okay. The failure of one or more chromosomes to align properly on the spindle causes mitosis to arise at the metaphase prior to the segregation of a newly replicated chromosome to the daughter nuclei. Wow. Okay, so I prepared only this much. I'll just stop.